In the second part of blood, we're going to talk about a couple of things. First, we're going to talk about clotting, then we'll talk about blood types and agglutination. Now, to begin with, hemostasis is what we call a blood clot. In essence, when we break a blood vessel, we have exposed connective tissue. Now, with the exposure to the connective tissue out here, it triggers the fiber response. Now remember that blood itself is also and so it contains living cells in the form of the erythrocytes, leukocytes, and platelets. It has non-living matrix, which is the plasma, and then we have the fibers, but the fibers are only seen when we have hemostasis, when we have to have some kind of coagulation. And it's interesting because remember that all of our vessels are lined with epithelial tissue, and that epithelial tissue literally acts as a blocker to the formation of these fibers. What happens when we talk about the formation of fibers is we're going to have a cascade event that triggers this until this area is stopped, and then normal functions can be restored. Now, the first step is platelet plug formation. In essence, the platelets, which are always in the blood, end up kind of getting sticky. And they start sticking to each other because of the collagen fibers triggering a chemical change in them. And as they stick together, they actually block whatever the break was in the lining or the vessel. And this is what creates the platelet plug. It's literally just a pile of platelets. Now you can see that here's the fibrin clot, that's this fiber that gets formed, but you can also see that there's these stacks of things underneath here, and that's actually our platelet plug. Now, the next thing after our platelet plug formation is going to be the vascular spasm. Now this kind of makes sense because we want to constrict blood flow to the area because we want to reduce the amount of blood lost. Keep in mind that the blood operates within a closed system. So it behaves in a fashion and maintains homeostasis, or all the homeostatic mechanisms behave in a fashion based on that assumption. And this is why if you have some kind of a really bad tear to an artery or a vein and you have a massive loss of blood, the heart rate tends to increase to maintain the blood pressure because there's a sudden drop in blood pressure. Since it's a closed system, the normal drop in blood pressure is often due to something like muscle contraction. So now we have a lot more blood flowing to the muscles, the blood pressure is going to drop, the heart rate speeds up, and this raises the blood pressure. Well, if it's no longer closed and we have blood flowing out, this is what gives us that positive feedback mechanism where we have more blood loss, the heart beats faster, increasing the blood loss, etc. So ultimately keep in mind that our spasms are narrowing our blood vessel, which will decrease our blood flow. Now the actual coagulation is a series of chemical reactions. These chemical reactions trigger enzymes. What's really cool about this enzyme cascade is that we have the event that's going to change A into this slightly different enzyme. Well, now that this exists, this is already in the blood and it acts as the enzyme for this event, creating a new enzyme here, which then acts as the enzyme for this substrate to produce this new substance. Ultimately, we produce what's known as fibrin. Fibrin is the stuff that comprises our blood clot. Now, it usually takes about 3 to 6 minutes. It remains until the endothelium regenerates, and it breaks down after the repair has been made, and then the body just starts breaking it down as a part of our tissue repair process. The main thing ultimately is we have this series of events. The one thing I definitely want you to know is that fibrin is what forms the meshwork, and that it's a series of enzyme or enzyme-mediated reactions that lead to it. One of the things you want to keep in mind is that as soon as this is triggered, it's basically a positive feedback mechanism until we form the clot. Then, once we form the clot, there's no longer the initial trigger up here, which helps to slow it down. Meanwhile, we have a process within our body that is actively breaking down fibrin. So as fibrin forms, we have another enzyme that's trying to break it down. And think about how important that is, because we do not want clots just to randomly form in our blood and run around and block blood flow to different tissues. Because again, without the blood bringing all that oxygen, all of the cells would die. And if a cell dies, the tissue is potentially damaged. If the tissue is damaged, the organ is potentially damaged. And all of this leads to this positive feedback escalation of events such that enough cells may die. If enough cells die in an organism, the organism could potentially die. You see this especially with blocks of blood flow to the brain. Remember when we talked about strokes in the nervous system? Those are basically a blockage of blood flow to the brain. The tissue dies, and sure enough, we might die. This is also what's behind heart attacks. Again, a heart attack is often the result of a blockage to an artery in the heart. The heart cells die, the whole heart stops working, and if the heart stops working, we no longer have blood flow. We've already talked a little bit about human blood grouping when we did the blood typing event, or the blood typing lab. So one of the things you want to keep in mind is that it is genetically determined, as we discussed. The antigens can be, the cell antigens are how the body knows whether something is foreign or belongs, and the antigens are what identify the blood cells in our bodies. Keep in mind that the antigens correspond to the opposite antibody. So if I had type A blood, I had B antibodies, anti-B antibodies. Type B blood, anti-A, etc. Now, let's go back. When we talk about Rh blood groups, this is the presence of what we call an Rh antigen. It's just what gives us the positive or the negative in our blood. Now, the last thing is, if we mix different bloods, we produce something known as agglutination, and this is really significant when we talk about the actual act of blood typing. By the way, this is what our blood typing activity should have looked like. And you can see the obvious clumping that's occurring due to the presence of the antibodies literally attaching themselves to the surface of the blood. And again, this makes sense in an immune system response because it makes it easier for our lymphocytes or the monocytes to be able to come along and phagocytize the foreign agent. Now, one of the things to point out too is that we have that agglutination is how we recognize our different bloods. This is how we cross type and match. And notice here and this is also what we talked about for a little bit, is we talked about universal donors versus universal recipients. This also ties into not whole blood. We're just dealing with the formed elements. So if we just take advantage of the formed elements, this is true. If it's the whole blood, keep in mind O can't be a universal donor because it has antibodies. This is why it has to be just the formed elements and not the whole blood. And then lastly, a quick reminder, if you have large losses of blood, again, this kind of goes back to what we were talking about. If you have this massive loss of blood, we have this massive change in how our heart behaves, which could exacerbate the initial stimulus. Transfusions are usually the only way to replace blood quickly. This is why oftentimes they try to do a quick type and match, and or it's good to know your type before you go into the hospital. This is also why a lot of people will bank their blood before surgeries.